I am your host, Zoya Rahim, and welcome to the 420 News. There has been a scientific breakthrough which scientists are calling DNA barcoding. DNA barcoding was first brought to attention in 2003 by Paul Hebert, a Canadian biologist and a professor at the University of Guelph. Today with us, we have the one and only Mr. Hebert to talk to us about DNA barcoding. Good afternoon, Paul.
Hello, Zoya. Thank you for inviting me on your show. It's a pleasure to be here.
No problem. Okay, so can you tell me why the interest in DNA barcoding?
Well, to be honest, 90 to 99% of fungi are being gone unidentified. This can cause unknown airborne fungi like mycotoxin producers.
Tell the viewers and I what DNA barcoding is.
Well, Zoya, a DNA barcode in its simplest definition is one or more short gene sequences that take from a standardized portion of the genome that's used to identify species through reference to DNA sequences. Barcodes are being developed for animals, plants, and fungi, but there's no general agreement to which region to use for protests.
So, Paul,
Yeah.
would you please tell us what steps are involved in DNA barcoding?
Well, Zoya, so as you like to say steps, I would like to prefer to say methods. So these methods include the latest information on techniques for generating, applying, and analyzing DNA barcodes across a tree of life, including animals, fungi, protists, algae, and plants. So you would first collect the specimen from the local environment and neighborhoods, then extract and purify the DNA from tissue or the processed material. Afterwards, you would amplify a specific region of chloroplast, mitochondrial, or nuclear genome by a polymerase chain reaction, and then after you would analyze that by gel electrophoresis. Then you would use the search tool BLAST and identify the sequence in database. Lastly, you would use multiple sequence alignments and tree building tools to analyze phylogenetic relationships.
Explain to me why DNA barcoding is being pursued by other scientists, not you. Other
scientists. Well, thanks for the question. I'd love to answer that. Thank you. So we'll start now. So, other scientists.
Yeah, other scientists.
Okay. So 1.7 million named species of plants, animals, and fungi are being pursued by international teams of scientists, as well as by groups and projects. Furthermore, I inspire the barcode movement and am a major participant in the eyeball project for scanning the life declared, and the approach is scalpel to the planet that by 2025, every species human encounter will frequently have been barcoded. And until recently, gene selection was largely restricted to easy to sequence and universal targets such as the ribosomal genes.
How does DNA barcoding affect the economy?
Well, Zoya, economically, land snails of South American forests are a source of food. And since ancient times, that has acquired new economic importance due to its cosmetic and nutritional properties. DNA barcodes of the species will help promote their sustainable use. Species substitution and mislabeling within fish supply chains occurs because of the price incentives.
How does DNA barcoding impact the society?
Well, hot topics for barcode researchers, policymakers, and government regulators involve consumer, agricultural, health, and environmental protection. So prominent human health-related efforts include barcoding several thousands of species of mosquitoes, including insects responsible up to 500 million human malarial infections and 1 million deaths per year. As well as malaria, mosquitoes transmit many other devastating diseases as well, such as the West Nile and dengue.
Okay, there has to be some negative things about it.
Unfortunately, there are.
Okay, so would you like to enlighten us about that stuff?
So if a taxonomist does devote extra curatorial and databasing effort to organize a museum's holding for barcoding, but only a small fraction of samples successfully sequenced, the negative psychological impact is huge. Equally bad is the damage and cost of having samples of a very large number of specimens, and a hope of a few of them will successfully sequence.
Paul, thank you for being on the show. It was a pleasure having you here. Unfortunately,
It was a pleasure being here.
Unfortunately, the segment has come to an end. Tune in tomorrow at 5 p.m. Central when Paul is gonna be back here tomorrow and he's gonna be talking to us about mitochondria. Till next time, good night, have an amazing day.